So coming to the second part of the cell cycle, that is regulation of cell cycle. Basically, cell cycle is a highly regulated process and it is regulated by large number of molecules, basically regulatory molecules, and they form a series of protein complexes. These signaling molecules or regulatory molecules, they are usually a series of protein complexes, and these protein complexes are made up of two subunits. They are made up of two subunits. One subunit is called as cyclin and other subunit is called as cyclin dependent kinase. That is CDK, cyclin dependent kinase. So it is basically a protein kinase. That is protein kinase. Cyclins, they are main regulatory molecules. They are main regulatory molecules. And they are also named so because they undergo a cycle of synthesis. They undergo a cycle of synthesis and degradation in different phases of cell cycle. In different phases of cell cycle. So that's why they are named as cyclins. It means their level fluctuates during different phases of the cell cycle, and on the other hand, these CDKs, they are catalytic in nature. They are catalytic in nature, but they do not fluctuate. Their level do not fluctuate, and they remain constant. They remain constant during cell cycle. They are catalytic in nature and remain constant during cell cycle. But these, for their catalytic activity, they are dependent on these cyclins, and when the level of these cyclins they increase or decrease, they also influence the catalytic activity of CDK, because these independently are neutral and they do not carry out any function, but when they form complex, then they regulate different phases of cell cycle. So it means cyclins, they activate the catalytic activity of CDKs. Basically these are dependent on cyclins, so that's why they are cyclin dependent kinase. And ultimately when cyclins they form complex, then they are active, and their main aim is to phosphorylate target proteins. That is phosphorylation of specific target proteins. Specific target proteins. So this is their main function. But how they carry out or regulate different phases of cell cycle, this can be explained as these cyclins, they are having three major classes. There are three major classes of cyclins. Three major classes of cyclins, and the cyclins which regulate G1 phase of interphase, they are collectively called as G1 cyclins, and second one is S phase regulating cyclins, they are S cyclins, and third one is mitotic phase regulating cyclins, and they are also called as M cyclins. So this is the table showing different types of cyclins which are found in different organisms, and there are different CDKs, cyclin dependent kinase, which are associated. So this represent cyclins and different CDKs with respect to different organisms. For example, in yeast Saccharomyces, there are different cyclin G1, S and M cyclins, and here in G1 they have cyclin 1, 2 and 3, and S cyclins it is 5, 6, mitotic cyclins they are 1 to 4. But one thing that is important to note down from exam point of view, all these cyclins of yeast cells, they have single common cyclin dependent kinase, that is CDC28, and it is also called as CDK1, and it is single CDK for all types of G1, S and mitotic cyclins. On the other hand, in vertebrates they have different cyclins. For example, G1 cyclins they are cyclin D, and it is having associated CDK, it is CDK4 and CDK6, it is CDK4 and CDK6. And in case of late G1 and early S cyclins, they are cyclin E, and they have associated CDK, that is CDK2. And S cyclins it is having cyclin A, and associated CDK it is CDK2. And mitotic cyclins they are cyclin B, and its associated CDK is CDK1. So here it is clear from your G1, S and M cyclins, that is they are in the sequence D, E, A, B, that is G1, this is late G1, this is S, and this is for G2, this is G1. And in mammals there are approximately 16 cyclins which have been observed, and there are approximately 9 associated cyclin dependent kinases which have been identified, and they are different from other vertebrates, and they share some common characteristic features in terms of the active sites on CDK and their regulation. So CDKs they are also called as cyclin dependent kinases. Basically they are a family of structurally and functionally related proteins. They are structurally and functionally related protein families, and they remain constant, as I have told you earlier that they remain constant during cell cycle, and their main function is they add phosphate group, that is PO4 group, to target substrates which are usually proteins. So their main function is they add phosphate group to target substrates, but for this their activity is regulated by CDKs. Their activity is regulated by the labels of cyclins. So cyclin label they regulate CDKs, and these CDKs they add PO4, that is phosphate group, to serine and threonine. Serine and threonine residues on target substrates or proteins. So this is important to note down. Cyclins, their level regulate. First, CDKs having catalytic activity, that is kinase activity, and this kinase activity add phosphate group to target proteins, and this addition it occur at specific amino acid residue sites, and they are serine and threonine. But the addition of this serine and threonine, uh, that is the phosphate group on these residue sites, it is on the target membrane, it is determined by the level of cyclins. So they are determined by specific, that is associated specific cyclins. Means cyclin of G1 can activate CDK of G1 phase, and then subsequently they can add phosphate group to serine and threonines of target proteins, which subsequently help in bringing the results or the products of G1 phase, and that is growth 1 phase, and in also in subsequent phases of cell cycle. And here this phosphorylation, it is not permanent type. Means if CDK has added a phosphate group to your target membrane or target protein, it means they are not supposed to remain same, as these phosphorylation they are transient and reversible. They are transient and reversible. Means once phosphorylated protein accomplished is its task, then it is eventually supposed to be dephosphorylated by another enzyme types, and these enzymes they are called as phosphatases. They helps in dephosphorylation. Of proteins, these target proteins, and this is also aided by, that is cyclin levels, they are also lowered, and ultimately Cyclin-CDK complex it is also disappeared. So first, phosphorylated target proteins, they helps in bringing or in completion of a specific task of cell cycle, and then subsequently this phosphorylation event is not permanent one, it is transient and reversible one. And here cyclins they are ubiquitinated, and CDKs or these your target proteins, they are dephosphorylated by other enzyme types in the cytoplasm, they are phosphatases, and that break down Cyclin-CDK complex as well as your target phosphorylated proteins, they are inactivated, they are also inactivated. Now important point is how Cyclin-CDK complex is influenced by cytoplasmic factors for its disappearance or inactivation, how it is inactivated or degraded. If this complex is there, then they will phosphorylate proteins, and ultimately they are supposed to repeat the same task, and cell is repeatedly undergoing same anabolic or catabolic event of same phase. but here these cyclins, their level fluctuates during different phases of cell cycle, so they are susceptible. And on the other hand, CDKs, they are also influenced by other regulatory events. But first is cyclin synthesis and degradation. Cyclin synthesis and degradation. The cyclin from Cyclin-CDK complex is subsequently ubiquitinated, that is ubiquitin protein is added to cyclin, and this cause the addition of ubiquitin or ubiquitination, it cause proteolysis of cyclin. So this ubiquitin is added to this cyclin by two major type of enzymes. One is called as ubiquitin ligase, and another one, it is also called as anaphase promoting complex. That is APC, and it is ubiquitin ligase, and it is also called as SCF, that is for its name, that is SKP1 choline F box, that is SCF, SKP1 choline F box. So this is ubiquitin ligase, and other one is anaphase promoting complex. So these are ubiquitin ligases that helps in addition of ubiquitin to the cyclin, and ultimately proteolysis of cyclin is achieved. But this ubiquitin ligase, it helps in destruction and degradation of G1 and S phase cyclins. They helps in degradation of G1 and S phase cyclins, ubiquitin ligase. And second one is this APC, anaphase promoting complex. It is responsible for ubiquitination and destruction of M phase cyclins, that is here ubiquitination and destruction of M phase cyclins. And due to these two events or their cumulative result, the cyclin level fall, and ultimately it cause inactivation of Cyclin-CDK complex. So this is first type of mechanism how Cyclin-CDK complex it is inactivated, that is why cyclin synthesis and degradation mechanism itself. And second is phosphorylation and dephosphorylation reactions. As you all know that the kinase activity of CDK, that is CDKs, they have kinase activity. it depends upon the phosphorylated state of catalytic CDK subunit. Means the phosphorylation state of this CDK, it is, and this phosphorylation dephosphorylation reaction, it is more relevant to fungus, that is S. Saccharomyces pombe. And here, in case of this is yeast type, here single CDK is responsible for the association with different cyclins, and different phases of cell cycle they are carried out. And here the kinase activity of CDK, it is influenced by phosphorylation. It is influenced by phosphorylation event, and this phosphorylation, it is basically induced by a factor that is called as V1WEE1. It is basically a protein tyrosine kinase. It is a protein tyrosine kinase, and it phosphorylate tyrosine residues on CDK subunit of complex, subunit of, that is Cyclin-CDK complex. When V1, that is a protein tyrosine kinase, it phosphorylate tyrosine residues on CDK subunit, then the result of this is it inhibit CDK activity. It means CDK is no more active for cell cycle. So V1, in case of yeast cell where single type of CDK, is responsible for different cyclin dependent cyclin complex formation. If it is phosphorylated by this protein tyrosine kinase B1, it subsequently inhibit CDK or its catalytic activity, and this phosphate here, that is phosphate, it is removed or dephosphorylated by CDC protein phosphatase. So it is dephosphorylated by CDC protein phosphatase, and ultimately it activates CDK. So here it is clear from this thing, if there is decreased V1 activity, and increased CDC25 activity, this cause, that is decreased V1 and increased phosphatase activity, it cause activation of Cyclin-CDK complex, and ultimately cell can proceed towards next phase. And the third mechanism for inactivation of CDK activity is binding of CDK inhibitor proteins, that is CKI. CDK inhibitor proteins, it inactivates Cyclin-CDK complex. This inhibitor molecules, when it associate with this Cyclin-CDK complex, there occur its inactivation. In mammals, there are two these CKIs, that is CDK inhibitor proteins, have been discovered, and they belong to two different broad families, that is CIP family, and it inhibit cyclin A and CDK2 activity. And second family is INK4 family, it inhibits cyclin D and CDK4 or 6 activity. So basically they are linked with inactivation of these Cyclin-CDK complexes in case of mammals. So from the above discussion, it can be concluded here in a brief manner in this diagram that different phases of cell cycle, they have associated different CDKs and cyclins. For example, in case of G1 phase of cell cycle, there are association of cyclin D and CDK4 and they form a complex. This is cyclin and its label fluctuates, and when this complex is formed, it activates target proteins and subsequently the growth it is achieved and maximum growth and size is obtained during G1 phase. And when this phase further proceed toward S phase, that is synthetic phase, then during this the first trigger is inactivation of this complex, and it is done by ubiquitination, and that ubiquitination is mediated by ubiquitin ligase. And this breaks down cyclin D and ultimately there occur dissociation and inactivation of this complex, and cell proceed towards next phase, that is S phase. Here different cyclin A, it approaches and form complex with CDK2, and this complex then allow different events that which are related with S phase, that is synthesis phase. But here the progression of cell cycle from G1 phase to the S phase, it is highly regulated one, because if in S phase there are no or insufficient raw materials formed which are needed for the replication and the transcription process, if they are not well maintained or synthesized or stored, then it can create problem. So here cell is first checked for all these events, and here it form G1 checkpoint, and it check whether cell has synthesized and stored all the raw materials which are required for DNA replication or synthesis phase. And in second phase, during S phase where DNA replication occur, here there is also another checkpoint, and it is called as replication checkpoint. Basically it check whether the DNA has been properly duplicated, and if there is any error, then it must be repaired and it should not follow the next phases of cell cycle. And but this replication checkpoint basically occur at G2 M transition, that is this is the main site where it actually arrest cell cycle to proceed further into mitotic phase. But important point from this whole process is that the entry and exit of different phases of cell cycle is mainly regulated by degradation and synthesis of cyclins, because here in this S phase, when cyclin A is degraded, then it proceeds toward G2 phase, and subsequently this replication checkpoint at G2 or M transition phase, when it allows its entry into M phase, here there is also another cyclin B, it is synthesized, and subsequently when it is degraded, this cyclin is degraded by anaphase promoting complex, and that is also a type of ubiquitin. It is also a type of ubiquitin ligase, and subsequently this degrade cyclin B and cytokinesis is carried out. So this is a whole process where different phases of cell cycle they are carried out. But let us discuss in a separate diagram about various checkpoints. So this is the diagram I have drawn here that represents various cell cycle checkpoints. So what are actually these checkpoints? Basically they regulate cell cycle and ensure that incomplete or DNA or damaged chromosomes, they are not replicated and passed on to the daughter cells. And in cell there are two different types of cell cycle, and they are called as fundamental checkpoints and additional checkpoints. So we have three fundamental checkpoints. First one is G1 checkpoint that occur at G1 phase of interphase, and second one is replication checkpoint. It is also called as G2 or G2M. It is also called as G2M transition checkpoint. And third one is, this is first one, this is second one, and this is third one in M phase, that is also called as mitotic or spindle assembly checkpoints, or also called as metaphase checkpoints. So these three, they are fundamental checkpoints. In G1 checkpoint, what happen? It basically check for nutrients. It check for nutrients, growth factors, and DNA damage. And here various mitogenic factors, that includes growth factors and other nutrients, they integrates with cell cycle and make decision whether to cell proceed, pause, or exit from the cell cycle. And if they are not properly maintained here in this phase of cell cycle, then they arrest the cell at G1 phase, and usually such cells they enter into a G0 phase. That phase is also called as resting stage. That is also called as resting stage. And when this checkpoint is crossed, the cell enter into S phase. And here in S phase, DNA replication occur, and subsequently cell undergo into next phase, enter into next phase, that is G2 phase. But here at G2 phase, at the end of G2 phase, there is also a G2M transition checkpoint. Basically it is a replication checkpoint. It operates at this transition from interphase into mitotic phase or the division phase, and it this G2 checkpoint or G2M transition checkpoint, it checks cell size, and DNA replication. So it has two major functions: cell size check and DNA replication checking, and it usually arrests cell at G2M transition, and thus it inhibit entry into mitosis or mitotic phase until DNA replication is complete or it is properly done. And in metaphase, there is metaphase checkpoint, or it is also called as spindle assembly checkpoint or mitotic checkpoint. So here it has two major functions. First, it monitors or check whether chromosomes and spindle fibers they are properly attached. This is first function, this checkpoint it ensure, and second, it prevent segregation of sister chromatids. It prevent segregation of sister chromatids until they are properly aligned at metaphasic plate. So it has two major functions. First, it monitor the attachment of spindle fiber to the chromosome, and second, it prevent the segregation of sister chromatids until they are not aligned properly at the metaphasic plate. So these are three fundamental checkpoints which ensure different activities in different phases of cell cycle, and there is additional checkpoint that is also called as DNA damage checkpoint. Basically, it operates at different parts of different phases of cell cycle. That it also operate at transition phase G1 S phase. It also operate in S phase. It also operate in G2 M transition phase. And main purpose of this is to ensure proper maintenance, repair of DNA of a nuclear genome. So these are different checkpoints. So there are three fundamental and one additional checkpoint. So from whole discussion that we have studied in this topic, interphase, it is the longest phase of cell cycle. Here it has G1 S and G2 phase, and it was first it was said to be the resting phase of the cell cycle, but in fact here there are a number of phenomena, there are a number of mechanisms that include DNA synthesis, growth, nutrients assembly, or cell size maintenance, and these all are carried out in this interphase stage of the cell cycle. So it is in fact metabolically most active phase of the cell cycle, and in fact it is not a resting phase. So this role of retinoblastoma protein, RB protein, and P53 protein. Basically, these are two major substrate or target proteins which are regulated by the complex of CDK and cyclins, and they play important role in cell cycle regulation. And as these RB protein and P53 proteins, they are collectively called as tumor suppressor proteins, and inactivation or loss of function mutations in these regulatory proteins can cause uncontrolled proliferation of the cells, and ultimately a cancerous growth can be developed. So here, coming to their major functions in the cell cycle regulation, first we will discuss these one by one. First is retinoblastoma protein and its gene. So this retinoblastoma gene, it is found on chromosome number 13, and it is having two copies in homologous pair of chromosome, and it expressed into RB protein, that is a functional regulatory protein. But loss of functional mutations in RB gene or its RB protein, they can cause retinoblastoma or other tumor types. For example, a childhood tumor of retina, which results due to inactivation of both the copies of which are found on homologous pair of chromosomes. If they are both inactivated or mutated, then it can lead to childhood tumor of retina. And these tumors, they are of retinoblastoma type of disorder. They are of two types. They are hereditary one, and second, they are sporadic in nature. And in hereditary, there is usually a defective copy of retinoblastoma gene is inherited, and they cause retinal tumors. And these retinal tumors, they are developed in early phases of life, and here both eyes are usually affected. But in sporadic disorder case, it is very rare. First thing, it is very rare, and they develop late in life, and usually one eye is affected. Here both eyes, they are not usually affected. And coming to its main function, how it works in association with Cyclin-CDK complex. So here Retinoblastoma protein, it is basically a substrate. It is a substrate for cyclin D. Cyclin D that is linked with G1 cyclins category, and this cyclin D CDK complex, and it exerts its effect during G1 phase. And here this retinoblastoma protein regulates the activity of a transcriptional factor that usually bind with DNA sequence and cause transcription of other type of cyclins, and that is cyclin A gene, and that subsequently lead to its transition from that is G1 phase to the S phase. But how it works, but how this RB protein, it bind with this E2F transcription factor and leads to activation or expression of cyclin A genes, and subsequently active cyclin A is synthesized. We will now coming to the regulatory function of RB protein, how it arrests the cell cycle at G1 phase. It has two scenarios. When there is no formation of cyclin D CDK complex, then it is inactive. But when cyclin D CDK complex is formed, it is activated one for this RB protein. And here they have two differential functioning with respect to this substrate protein for this complex. And here when there is no active cyclin D CDK complex, there occur no phosphorylation of this RB protein. And when this protein is not phosphorylated, it binds with E2F transcriptional factor, and this binding induces suppression of the activity of this transcriptional factor, and here it is incapable of binding with the DNA sequence of cyclin A, that is cyclin A gene, and ultimately no active synthesis of cyclin A or S phase gene expression occur, and thus cell cycle is arrested. But when this cyclin D CDK complex is formed, it is activated one, and here it causes phosphorylation of retinoblastoma protein, RB protein, and this lead to reduced affinity of its binding with the E2F transcriptional factor, and this reduced affinity, it ultimately allow this E2F factor to bind with DNA sequence on cyclin A gene, and ultimately its expression lead to synthesis of S phase gene products, which are here cyclin, and with that cyclin is cyclin A type, and ultimately cell enter into S phase. So from exam point of view, here E2F, that is a transcriptional factor, it is always able to promote S phase, but here RB protein, it is regulatory one, and it can arrest cell in late G1 phase if it is phosphorylated and if it is not phosphorylated. So here this phosphorylation, it cause the expression of cyclin A proteins, but when there is no phosphorylation of RB protein, there occur no synthesis of cyclin A. So now coming to the next protein, that is P53 protein, and how it regulates cell cycles. So first starting this P53, it is a tumor suppressor protein, and it arrests cell at damaged DNA. Arrests cell with damaged DNA at DNA damage checkpoints. Basically how it works, with the functional P53 protein, it arrests cell usually at G1 and G2 phase. But when cell lack functional, there is absence of functional P53 protein, the cells are not arrested at G1 phase, and it can lead to uncontrolled proliferation of cells, and that ultimately cause cancerous or tumor type growth. So coming to its origin, it is encoded by a gene, P53 gene, which is present on chromosome number 17, and this cause expression of a P53 protein that usually have 375 amino acid long sequence, and this polypeptide, that is P53 protein, act as a transcriptional factor which regulates the expression of cell cycle proteins. So in normal case, what happen? This P53 it is not required. This P53 it is not required in normal conditions, and it is extremely unstable. And how it is made extremely unstable? Because in normal conditions it is recognized by a type of ubiquitin ligase, that is MDM2. It is MDM2 ubiquitin ligase, and this bind with this and cause its degradation. So it is clear in its function of P53. In normal case, there is no DNA damage. We know that that's why it is a normal one. And here P53 protein, they are ubiquitinated by MDM2 ubiquitin ligase, and they are degraded. And their levels, they are maintained at very low levels. They have low levels of P53 protein. But when there is a damaged DNA, here comes the main role of P53 protein. When there is damaged DNA, it causes activation of protein kinases, and there are two types of protein kinases, ATM and ATR. These are protein kinases, ATM and ATR. There are two types, first and second types. ATM stands for ataxia telangiectasia mutated type of protein kinases. And these protein kinases, basically they are sensors. They sense something, that is DNA damage or defects. And here this ATM, it is a sensor for DNA defects which are caused by ionizing radiations. It is important from exam point of view. They act as sensors for DNA defects which are caused by ionizing radiations, while ATR, they are specialized for detection of DNA defects which are caused by either UV rays or by the inhibitors of DNA replications. They are non-ionizing radiations and inhibitors of DNA replication. So main mechanism how these ATM protein kinases, they exert their role on these P53 regulation, because we know in normal condition they are ubiquitinated and they have low levels of P53 proteins. But when there is DNA damage, activation of ATM protein kinase cause phosphorylation. Here these 53 proteins, they are phosphorylated, and this phosphorylation, it alter the site on P53 protein where ubiquitin ligase, that is MDM2, it bind. So basically it prevent the formation of P53 MDM2 complex. So this complex, it is not formed, and here the level of P53, it goes on increasing, and this increased P53 level, it arrests cell at G1 phase. If it is lower, it is having P53 having lower level, then cell enter into next phase. But if these levels, they are increased, then cell is supposed to arrest at G1 phase. And when this DNA damage, it is repaired, then after repair, cells can normally divide and can complete its cell cycle. So this is all about cell cycle regulation and how it is regulated by different regulatory proteins at different phases of cell cycle.
If you like my video, you can share it, comment on it, and can subscribe my YouTube channel for many more such videos. Thank you.